So today I'm going to talk about some work that I did during my PhD about studying metabolic diversity and then trying to use the enzymes as tools to enhance plant metabolic diversity. So it's nice to see some names that I recognize pop up in the chat. I also invited my biochem class to join us today, and I saw at least one from biochem class, so I think that's fantastic. So yeah, hopefully this is not too crazy long, and we can have a nice discussion afterwards. So I think this is a great seminar series. So Gaurav, thank you, and also Lars and Luke, thanks for organizing and inviting me, but also just getting this up and going. I think it's a fantastic venue. Okay, I don't think I have to say much about this, but I will just say briefly, plant chemicals serve as the language of life. I think we all know this. I'll just say a few brief things. We all know that plants are amazing chemists. They've developed a structurally diverse chemical arsenal to enable environmental interactions, both defensive but also attractive properties of specialized metabolites. The other aspect of plant specialized metabolites is that these have often been co-opted by humans, where they can serve nutritional, industrial, or medicinal roles. So there's many aspects that have broad implications for plant biochemistry. So what I'm particularly interested in, let me get my laser pointer up and going, is really what are the underlying biochemical mechanisms that enable this plant chemical diversity. And if we know more about these underlying biochemical pathways, enzymes, reaction mechanisms, what have you, can we use these and harness these to engineer more resilient crops or to be used as tools in synthetic biology platforms? So I use the rapidly evolving plant specialized metabolic pathways as a model system to essentially address these questions. All right, one last unnecessary slide about plant specialized metabolites. These are usually biologically active end products. They are distributed in a lineage specific manner. So we know that many of these compounds are restricted in their phylogenetic distribution. And finally, they're often produced in cell or tissue specific types. There's many examples here. And so it's these characteristics that actually used to hinder the study of plant specialized metabolism. But with the advent of cheap sequencing and comparative genomic approaches, it's actually the lineage and tissue specificity that it is actually facilitating the discovery of plant metabolic pathways. And there's countless examples of people putting biochemical pathways together left and right these days, aided by comparative approaches and cheap sequencing. Okay, so the specialized metabolic or the class of specialized metabolites that I've been using to explore the evolution of chemical diversity and also to use these as a source of diverse enzymatic activities are acylsugars. These are a class of defensive compounds and sticky compounds that are found throughout the Solanaceae. And they look like this. They typically have a sugar core. It can be a disaccharide or a monosaccharide. And on the hydroxyls, there are acylations, which can vary in length, branching pattern, and also position of acylation and actually number of acylation. So as I mentioned, they're fairly ubiquitously distributed across the Solanaceae family. They are synthesized in the glandular tip cell of trichomes, and they have defensive roles, both in direct defense and indirect defenses. Okay, so a little bit about their biosynthesis. Acyl sugars are built from simple core metabolites. The acyl chains are primarily derived from the branched chain amino acids, and their sugar cores are coming directly from sugar metabolism. The acyl chains are activated as CoAs, and they can be esterified onto these hydroxyls by a class of enzymes that I'll refer to as acyl sugar acyltransferases. And these can add acyl chains in various positions, leading to diverse acyl sugar products. Another mechanism that enables more diversity of structures is that these acyl chains can be elongated, which leads to a diverse pool of acyl CoAs in the trichome tip cells. And then these can be esterified by ASATs onto sugar cores, leading to acyl sugars with varying acyl chain length, varying branching patterns, varying positions on the sugar core, and actually numbers of acylations. And so if you look within a single species, there can be diverse acyl sugar variants. But if you look more broadly across a genus or even the entire family, there's tremendous acyl sugar structural diversity due to the mechanisms that I just alluded to. So it's the trichome specific accumulation and biosynthesis together with diverse acyl sugar products within a species or across the entire family that make this an excellent system to explore the evolution of specialized metabolism. And so today I'm going to talk about using the Nicotiana genus, which is in the Solanaceae family, as a source for diverse acyl sugar variants, but also diverse enzymatic activities. Okay, so we took a kind of a multi-pronged analytical approach to discover acyl sugar structural variants. And we did this across the entire Nicotiana genus, which is a genus of around, I don't know, 50 or 60 species. Since these are produced on the trichome tip cells, their extraction is really straightforward. You just cut off a leaf and dip it in solvent, and then you could analyze that using either LCMS, GCMS, or any other analytical tool. I'll primarily talk about LCMS today, but I won't talk much about details. So an undergrad who worked with me at the time, who was an REU student for the summer, Mackenzie Jacobs, did all this work. So she grew out all the Nicotiana, did the acyl sugar extractions and LCMS, and these are just some representative chromatograms from varying species. Oops. So you can see that there's peaks with different retention times. This suggests different acyl sugar variants, either due to differences in core or acylations. You can count all these acyl sugars up, and then you can look at their fragmentation pattern to get an idea of the structures of these compounds. And so you can do this across 60 or so species, and you're going to get a lot of data. And that's exactly what Mackenzie did. And to make a little more sense of this, or to derive some patterns, what we did is we took the fragmentation patterns and we mapped the acyl chain types into a phylogenetic context to see if we could identify any patterns. And this is what we get. So these are all the species that we screened, and red boxes indicate that that species had an acyl sugar with that acyl chain type, and white boxes indicate that those species didn't have that acyl chain type. So a few things to note is that Nicotiana didn't make acyl chains longer than 8 carbons in length. This is in stark contrast to other Solanaceae plants that can accumulate acyl chains up to 14 carbons long. Another thing to note is the C5 acyl chain was fairly ubiquitously distributed. The longer CoAs were a bit more sporadically distributed. And there was this one particular CoA that I'm calling 5D here because it was 2 mass units less than a typical C5 acyl chain. So this indicated a desaturation. And this was restricted to a few closely related species. So we wanted to look a bit more into these closely related species. So we did GCMS analysis of these acyl sugar extracts to get a little bit better idea of the structure of those particular acyl chains and their relative composition. So what we did was we converted the acyl chains into fatty acid ethyl esters and then analyzed those using GCMS. And so what we found is that that 5D acyl chain was in fact a desaturated 5-carbon acyl chain that I'll either call Tiglyl-CoA or tiglic acid. And in this one particular species, Nicotiana acuminata, this acyl chain accumulated upwards of about 75% of the total acyl chains on acyl sugars. So this was quite intriguing to us. Then we also wanted to resolve the structure of the putatively tiglated acyl sugars. So what we did was we grew up a lot of Nicotiana plants, extracted those, ran out the acyl sugars on a long gradient and isolated 2 acyl sugars. And Talani Anthony, who's a postdoc at Michigan State, solved these structures using NMR so that we could actually get the real structure. And these 2 acyl sugars indeed had the tiglyl acyl chain. They actually had 2 of them in the same positions. The only difference between these 2 acyl sugars was this one had 4 acylations in total, whereas this one had 5. The only difference being the presence of this acetyl group at this position here that's not here. And so the presence of this uncommon acyl chain and its abundance led us to want to identify the genes that were involved in its biosynthesis. So we focused on pathway discovery in Nicotiana acuminata because of the accumulation of this very uncommon acyl chain. And so we did a comparative transcriptomics approach, taking advantage of the fact that these are trichome localized metabolites. So this is a stem of Nicotiana acuminata. You can kind of see it glistening in the light. That's all the sticky acyl sugars. So what we did was we scraped trichomes from the stem and compared that to the underlying shaved stem tissue. And then we did a de novo assembly and then looked for candidate genes that were enriched in the trichomes. And we used biochemically characterized ASATs from tomato and other species. And we were able to identify 4 candidate enzymes. And the next step was to functionally validate these candidate genes. And so we took a few different approaches. This is what the pathway looks like in tomato, where you have an ASAT1 that acts first. It uses acyl-CoAs and sucrose to make a monoacylated sucrose. And then the other ASATs act sequentially, and you can make a tetraacyl sucrose. So we assumed that the Nicotiana pathway would be similar to this. And so we started actually with an ASAT1 homolog in Nicotiana and an ASAT4 homolog. And we started with these because they're a little bit easier to assay in terms of getting the substrates, because you can get CoAs and sucrose from sigma. Whereas the pathway intermediates don't typically accumulate in plants, so they're a bit harder to access. And we could test the ASAT4 activity because we had those 2 NMR resolved acyl sugars in hand. So we started with ASAT1 first, and I used a gene silencing approach to try and validate the function in planta. So I used VIGS to do this. And compared with an empty vector control, using 2 different VIGS fragments that targeted different regions of ASAT1 gene, their acyl sugar levels were significantly reduced compared to an empty vector control. So highlighting or suggesting a role in acyl sugar biosynthesis. So the next step was to validate these in vitro. And so we purified ASAT1 and then tested it with sucrose and IC5-CoA, and then monitored the reaction for accumulation of a monoacylated product. Just to reiterate, this is a highly trichome expressed gene. And so in that assay, we identify a monoacylated product, monoacylated sucrose that's absent in the negative control. So this kind of reiterates what we found in the in vitro or in the in vivo silencing approach. And I did kinetics, and this enzyme behaves similarly to previously characterized ASATs for the CoA donor and the sugar acceptor. So given though that we detected a diverse array of acyl chains, not just C5 acyl chains, we wanted to test if ASAT1 could test could use a broader diversity of acyl donor substrates or CoAs, or if it could also use a diverse array of acyl acceptor substrates. So we tested first the acyl donor promiscuity. So we tested from benzyl CoA, which is an aromatic CoA, all the way to a 14 carbon long acyl CoA. And ASAT1 can use all of them, but to varying degrees. It likes the longer and the straight CoAs. So this highlights the promiscuity that ASAT1 has for the acyl donor. So then we tested acyl acceptor, and it was much more specific for the acyl acceptor. So we tested it with disaccharides like sucrose, and also some monosaccharides. I'm only showing the disaccharides here. So it used sucrose. It also used trehalose, but about 50-fold less active with trehalose than sucrose. But it didn't use other disaccharides, and it wasn't able to use monosaccharides. So this highlights the specificity it has for the acyl acceptor. So then we wanted to test ASAT4. And like I mentioned, we could take advantage of those purified acyl sugars that we had in hand to test this. These are what those two acyl sugars look like from their retention time from a Nicotiana acuminata acyl sugar extract. So then we set up an enzyme assay where we used the acyl sugar with 4 acylations. We added acetyl-CoA and a purified ASAT4, and then monitored the reaction for an acyl sugar with 5 acylations. And that's exactly what we got. So this is a peak that corresponds to the substrate, the S4, and this is the peak that corresponds to the product. And so this demonstrates the function of ASAT4, also showed the function of ASAT1, but it's the middle steps that can be a little tricky. And this is where we kind of switched to a in vitro pathway reconstruction way to validate the function of those intermediate steps. And so essentially, that's just putting all your candidate enzymes in one reaction at a time. And so that's what we did. So we added ASAT 1 through 4 purified enzymes together with CoA substrates and sucrose, and we monitored the reaction for higher acylated acyl sugars. And so I'm showing you this kind of in a stepwise fashion. So if we added ASAT1, we get production of a monoacylated sucrose. If we add ASAT1 together with 2, we get production of the monoacyl sucrose, but we also see appearance of a diacyl sucrose product. If we then add in ASAT3, we start to see a triacylated product. And then finally, when we add ASAT4, we see all the way up to an acyl sugar with 4 products. So this is a simpler way to look at the data. The assays are a bit more complex in terms of products that are produced. So this is actually what the chromatogram looks like. And we were able to annotate these peaks based on accurate mass and fragmentation pattern. So we see a monoacylated product. We actually see 2 diacylated products, 2 triacylated products, a product with 4 acylations and one with 5. So this kind of circumvents the difficulty of obtaining the substrates in the middle of the pathway, and it also demonstrates the function of these four genes. or the 4 enzymes. And so given the promiscuity of at least ASAT1, we wanted to test if we could create more structurally diverse acyl sugars by mixing and matching acyl CoAs. And so what we did was we replaced the C5 CoA with NC6 CoA. And these chromats are a bit busy, but I'll try my best to walk you through it. But essentially, we see all the same products as we did when we used the C5 substrate, except that a C6 acyl chain has replaced that. So we see all the way up to a pentaacylated sucrose. When we do the same thing, but we add a C8 CoA, we get all the way up to a pentaacylated sucrose. So this can make a diverse array of acyl sugar products with these 2 substrates. However, as we increase the acyl chain length, we see fewer and fewer products. So when we use NC10 CoA, the highest acylated product that we see is a triacylated product with 3 10-carbon acyl chains. And when we use C12 CoA, we only see up to a diacylated product. So this highlights that as you increase the acyl chain length, this appears to be a limitation of these ASATs. Is they're promiscuous, but as the aliphatic chains become larger, maybe these substrates and products can't be accommodated in the active site. However, some of these products do not accumulate in plants. For example, all the products with a C10 CoA and all these products with a C12 CoA. So this demonstrates the untapped potential of these enzymes for synthetic biology platforms. Okay, so we took it one step further. Geez, are people seeing a really crazy screen?
Yeah, I think maybe just go forward and back like one more time.
Okay.
It's like a very complex heat map.
It's not a heat map, but let's see if I can go. Let's see. Okay, here we go. I think it's just a heavy slide. Okay, so we took this one step further and we mixed and matched a bunch of acyl CoAs just to see if we could produce lots of acyl sugar variants in a single in vitro enzyme assay. So we used all these CoAs together with our 4 ASAT enzymes and sucrose. In summary, we can make almost 30 acyl sugar variants in this one enzyme assay. The data is a little hard to look at because a lot of these acyl sugar variants are very similar and hard to separate using chromatography. And so you can look at these kind of individually. So these are the monoacylated products, diacylated products. I'll show all of these, though it's probably not even necessary. But the takeaway message is that when we mix and match CoAs in this in vitro platform, we can make upwards of 30 acyl sugar variants, which is actually beyond the diversity that we see in a Nicotiana acuminata plant extract. So the fact that we can make more products in vitro than are present in planta suggests that the bottleneck in plants, or the metabolic constraints perhaps, is actually the availability of CoAs in the trichomes, and that we might be able to enhance the diversity of acyl sugar products by tweaking the CoA pool in the trichomes, and this may require manipulating core metabolic pathways that sit upstream of ASATs. And so mixing and matching of CoAs was a nice way to make diverse products in vitro. We next tried to mix and match ASATs from diverse Solanaceae lineages to see if that might be a good approach to make diverse acyl sugars. In short, it didn't work as well as we had hoped. But one thing it highlighted, and I'll allude to this, is maybe how the pathway has evolved in some of these species. And so I'll just touch on actually two assays that we did. So we combined Nicotiana ASAT1 with a tomato ASAT1. So these are both within the Solanaceae, but a bit divergent. And we didn't expect much from this assay other than maybe getting a monoacylated product. But in fact, in addition to a monoacylated product, we actually also saw a diacylated product. So this was quite intriguing, that if you add 2 ASAT1 enzymes, you can make a diacylated product. So we wanted to know, which of these ASATs is actually doing the second reaction. So we set up sequential enzyme assays. This is the assay if you just combine Nicotiana ASAT1 with tomato ASAT1. So you make monoacylated products and diacylated products. And I switched the column chemistry up here, sorry. So your monoacylated products here now on the right, and the earlier eluting peaks are your diacylated products. When I set up an assay where you add tomato ASAT1 first, stop the reaction, then add Nicotiana ASAT1, I only detected monoacylated products. When I do the reverse, where I add Nicotiana ASAT1 first, stop the reaction, then add tomato ASAT1, not only do I get monoacylated products, but I also get diacylated products. So it seems like ASAT1 can add first, followed by tomato ASAT1 can add second. So the question though really is, where does this ASAT2 activity come from on tomato ASAT1? And so I'll try and briefly summarize what I think. This is a very busy slide, but the top is the tomato pathway. The bottom is the sister lineage pathways represented by Nicotiana petunia or salpoglossus. So the ASAT1 from Nicotiana, which groups within this blue clade, which is this clade that's the sister lineages to the solanum. So all these lineages have an ancestral, what we're calling an ancestral ASAT1 sequence. So this is the one that can add an ASAT or add an acyl chain to a sucrose to make a monoacylated product. Well, tomato doesn't have an ortholog in this clade. So somehow this gene was lost in the solanum lineage. ASAT1 sequence, the one that can catalyze this first step in the reaction, groups within a clade that contains ASAT2s from this sister group. And so it's likely that tomato ASAT1 evolved from an ancestral enzyme that had ASAT2 activity. This ASAT2 activity was retained on tomato ASAT1, but then somehow it evolved ASAT1 activity de novo. And this may suggest kind of how that pathway evolved and why there's slight differences in various Solanaceae lineages. And it's possible that we see similar mechanisms occurring in the other steps of acyl sugar biosynthesis in other species. Okay, so in conclusion, I showed you that tremendous acyl sugar structural diversity exists across the Nicotiana genus. I've shown you that in vitro pathway reconstruction can expand upon acyl sugar structural diversity, and it highlights some of the promiscuity of these ASATs, but also metabolic constraints of Nicotiana acyl sugar pathway. And by combining ASATs from diverse Solanaceae lineages, sheds light on acyl sugar pathway evolution and mechanisms that potentiate chemical diversity. So with that, I'd like to thank the NSF for funding this project, Rob Last, my postdoc advisor, the last lab, all the members of the last lab, undergrads who helped with the project, and also collaborators at Michigan State. And I'd be remiss if I don't use this opportunity to promote my own group. Like Gaurav said, I just started my own group at the University of Missouri, and I'm looking to expand upon the lab. Right now, it's me and 2 undergrads, and we're looking to hire additional people who are interested in discovering metabolic and chemical diversity. So if this is interesting to you, please reach out to me. And thank you all for your attention.
Thanks, Greg. So
Should I stop sharing or leave slides up?
it's fine if you keep it on. There is a question in the chat.
Okay, let me look at the chat. Okay, an odd chain fatty acid common in Nicotiana. Are these derived from fatty acid synthesis process? That's a good question. We don't know a lot about how acyl chains are elongated. So that is one focus of my current group. It is likely that some lineages, for example, tomato and closely related species, do use a fatty acid synthesis like 2-carbon chain elongation mechanism. So what we find is some of the fatty acid biosynthetic genes are highly enriched in the trichomes, and there seems to be gene duplications and co-option of these genes. In Nicotiana, it's more likely that they use a different elongation mechanism, something that's a bit more analogous to the citric acid cycle. We really don't know anything about it beyond that. It could be plastidial, it could be mitochondria. ASATs are 99.9% sure they're localized to the cytosol. That's another thing we just don't know much about, is how this subcellular interplay or architecture it works in this pathway. I'll just keep going on the chat. Sandra says, could you elaborate a little on the linearity of the pathway? Is the pathway really a linear one? It's a great question. Let's see. I think it's somewhat linear. So for example, if I do, when I do an enzyme assay that has acyl-CoAs, sucrose, and ASAT1, I make a monoacylated product. But if I use any of the other Nicotiana ASATs to try and force them to do the first step in the pathway, they don't do it. So in that sense, there is an order to these reactions. But I also showed you, let's see, maybe I can go back to that slide. I also showed you that, oh geez. For example, here, you get a diacyl sucrose that has an acetyl-CoA, or a 2-carbon chain and a 5-carbon chain. The enzyme that adds the 5-carbon chain we call ASAT4. But here it looks like it can use only a monoacylated sucrose. So in that sense, it may not be linear. Again, this is an in vitro reconstruction. So this isn't a great proxy for what's actually going on in the trichomes. And we do know of mechanisms that occur in the trichomes that may be recycling, or essentially being like a quality control mechanism that may eliminate these off pathway products. I don't know if that's a great way to describe them, but I think your question is very good. I think it's probably mostly linear, but there's possibility that it diverges at some branches.
Hey, Greg, maybe I can just like go in here. So could it be possible that this product, which you just elaborated on, is then like furthermore converted because other enzymes also would take that? Like that it's more like a grid than a linear one, right?
It very well could be,
Yeah.
yeah. I mean, it's easiest to draw it in a linear fashion. I think the best is to knock out these enzymes one by one using CRISPR and see. But yeah, I think it's probably acting more like a grid-like rather than a linear. But there is kind of an order of these acylations.
Yeah, could you test that with like other tobacco varieties where, you know, it stops like at that point or it doesn't have that enzyme and just like transiently express additional enzymes and see what the outcome is? Were you thinking about that?
That's a possibility. So you're suggesting that and maybe other tobacco species that may only produce diacylated products, say, adding in additional ASATs to see if you can get additional product diversity in an implant system.
Yeah.
Yeah, I think that's a great idea.
Cool. Keep me updated. Thank you.
You're welcome.
Another question from Luke.
Oh, okay. So Luke asks whether ASATs interact physically. This is something I would really like to go after. There's no indication that they do, but the fact that you don't see pathway intermediates accumulate in plants at all suggests there could be physical interaction in terms of a complex or a metabolite. It may suggest that downstream enzymes are just extremely efficient. I don't know that I buy that for ASATs because we know that they're not extremely specific either. So it could suggest that they're forming some sort of complex in the trichome. So I would like to try and pursue this. I don't know, easier said than done, but I do think it's worth looking at. So it's a good question, Luke. I don't know if we should go to Benji or stay in the chat.
I think Benji raised his hand first. Benji and Todd, and then Lars.
Great. Thanks very much. Yeah, thanks, Craig, for a really interesting talk. I liked what you said about the idea that some of these acyl groups are kind of just, you know, when there's an abundance of them from primary metabolism, they might attach to the, you know, be preferentially attached to these sugars. So I'm wondering kind of if you can expand on that. And do you think that the diversity is driven by, you know, diversity and sort of, you know, interactions between the plants and kind of pathogens or herbivores, or do you think it is kind of just like metabolic overflow from primary metabolism that causes these changes in lineages?
Yeah, I can speculate, but I don't have a great answer. I mean, those are really good questions. If I extrapolate the in vitro reconstruction, I think the limitation of product diversity is the CoA availability in the trichomes. Given how promiscuous these seem to be in vitro, I would expect that they're acting just as promiscuously in the trichome. So my sense is that the limitation of product diversity really lies in the CoA availability, and that rests upstream in core metabolism. So I think, you know, an easy way to maybe test that would be, can you manipulate core metabolism in, say, Nicotiana to make longer CoAs in the trichome tip cell? Then would they then be able to produce acyl sugars with long acyl chains? My sense is yeah. I could be wrong. In terms of relating this back to biological implications, I think at some point the diversity of acyl sugars is probably also driven by pressures from the environment. But I can't really speculate more on that. I wish I knew more. I'm not a chemical ecologist.
Can I jump in, Craig? On
Yeah,
that?
go for it, Gaurav.
Yeah, so Ian Baldwin's lab has done beautiful work in 2012, 2014, where they showed that some of these acyl chains, at least in the Datura, are really important from an ecological perspective in terms of modulating diatrophic interactions. But some of these species produce 800 acyl sugars on a single leaf. And it's not, I mean, each of those acyl sugars having function is probably not something that might realistically happen. So there is, I think, a large element of promiscuity of these enzymes. These are members of the large acyltransferase family. So there's a substantial contribution of promiscuity that produces this diversity.
Great, that's really interesting. Just to follow up on that, just briefly, is you kind of this Tiglal acyl group is pretty interesting. So is there a specific biosynthetic? Is that a general metabolite? I don't know, or is it is it specific in that lineage?
It's actually a breakdown product of branched chain amino acid. So when they get broken down, this is an intermediate in that that breakdown step. So my hypothesis is that one of the genes that's responsible for branched chain amino acid catabolism is not trichome expressed. And so you're leading to this accumulation of this catabolic product. And so you have Tiglal-CoA in the trichome, so ASATs are going to use it.
Yeah, great. That's a really nice hypothesis. Thanks.
Should we go to Todd?
Oh, thanks for the interesting seminar, Craig. So I was interested in the kinetic data you showed. And I don't know if that slide's still easily accessible, but the Km for sucrose looked pretty high. And so one thing I've been wondering lately is because it's based on what other people say to me when I show our kinetic data, and they say, God, your enzymes suck at what they are supposedly doing. And so, so at any rate, I can imagine in a trichome sucrose concentrations might be super high. I would be a little bit more surprised maybe if the monoacylated ever reach, or diacylated products ever reach such high concentrations. But I wonder if maybe we shouldn't be thinking about secondary metabolism in terms of what the Km actually is, because plants have days and weeks to make products, and even if they're pretty slow, eventually it all works out. So do you have any perspective on any of that? Yeah,
so good eyes. I thought maybe only Gaurav and Lars would notice these crappy KMs. So yeah, the KM for sucrose is about 5-10-fold higher than actually tomato ASAT1, and some other biochemically characterized. So even if it were 10-fold less, it's still on the order of 5 millimolar. I mean, that's still kind of a cruddy KM to begin with. So I don't know. I like the idea of what you're saying, that these plants have time to make these compounds, so you don't necessarily need to be the most efficient enzyme. Though when I do these assays in vitro, I'm only incubating these for 30 minutes. And so I think they're pretty efficient. I'm also using a really high sucrose and CoA concentration. But we really don't know about the concentration of these compounds in trichome tip cells. I've tried to measure the CoA pool. It's just not that straightforward. These are hard things to extract because they're quite reactive. So that it would be nice to know the concentrations of CoAs and sugar acceptors in the trichome tip cells. We don't have a good sense for that.
Thanks.
Should I go back to the chat?
Go ahead.
Okay, I'm just scrolling up to see maybe where I left off. Oh, Lars's question. Yeah. Okay. Are ASATs always strictly expressed only in the trichomes? No, they aren't. Sometimes in the roots. Are there ASATs not expressed in the trichomes? Yes, the ones in the roots. If the enzymes are promiscuous, expressing them in different cell types would probably lead to many new metabolites. I totally agree. So the lab is the last lab, not my group, is currently deep into a root-specific acylsugar biosynthetic pathway in tomato. So, if we move these trichome-expressed ASATs into the roots, maybe we would see differences. You do see different types of acylsugars produced in the roots. I don't know if that relates to promiscuity of these, or the substrates available in the roots are quite different from the substrates that are available in the trichomes. I think that could be another good test. Take a trichome expressed ASAT1 and put it in the roots, and then do you see any new products? If you don't, then it's probably the products you see is probably due to the pool of CoAs available to those ASATs.
Yeah, I was also thinking that I must have done like very strict regulations. Like I remember this one from like someone earlier publication from the last lab, where they have these very green fluorescent tip cells in the trichomes, and I was wondering if all of these ASAs are really that tightly controlled under some promoters to make sure that they're not like expressed in some other tissue where they would like produce any compounds that might be detrimental to the plant.
All I can say is that they are pretty specifically expressed in the trichomes, though they do show some expression elsewhere. But then we find examples of those that are specifically expressed in, say, root. I don't know if it's root hairs or root something or others. I was looking to see if someone from the last lab was on that could comment on that. Paul was, but then he may have dropped off.
Okay, thank you.
Maybe we go to Ryan and then back to the chat.
Hey, Greg, good to see you, and thanks for the great talk. I was wondering if you could give your thoughts or elaborate / speculate a little bit on, after studying a bunch of these ASATs, both your work and the collective work from the last lab, have you gained enough insight, would you say, to leverage either the promiscuity or the specificity of some of these ASATs towards having regio or substrate, like acyl substrate specificity for generating these acyl sugars? Like, do you think that's something that's approachable in the near future, or is it just a little bit too unpredictable?
I'm not sure about predictability. It's a good question. I think we have enough activities and diversity and sequence where we could correlate, particular amino acid residues with specificity or regio specificity, like you said. That's the one thing about ASATs, is they're very specific to the hydroxyl that they acylate. I do feel like that is something we can kind of figure out the molecular mechanism for. Why does an ASAT1 always add at the R2 position? What is it about the active site that enables that? It would be helpful if we had a crystal structure. These aren't the easiest enzymes to express and purify. I think someone is probably, or this could be a good project for someone to work on. Yeah, crystal structure would help. It's not a great answer, but it's good to see you too, Ryan.
Yeah, thanks.
Should I go back to the chat?
Yeah, question from Marcos.
Okay, good to see you, Marcos. If you remove any of the downstream enzymes, does the reaction still work? if you remove ASAT2 and keep, yeah, so we did that. Good question, Marcos. And this kind of goes back to Sandra's question. Is it more grid-like or linear? So in your example, if I do ASAT1, 3, and 4, I only get a monoacylated product. So it kind of breaks down. So, you know, these are acting sequentially in some sense, but I guess I should amend my statement. If I add ASAT 1, 3, and 4, I could get a diacylated product, something with an acetyl group and a long chain. But this isn't then the substrate for, say, ASAT3. So yeah, it's partly linear and partly grid-like in that sense.
A couple of questions about functionality. About functional differences, whether the acyl chains have.
Of course, yes, but I'm not the one to talk about that. But you know, if you isolate acyl sugars with different acyl chain lengths, these will have differing effects on different types of organisms. There are definitely biological implications for some of the product diversity that is observed. Anything else to say on that? It's not really my area of expertise, but yeah, there's certainly, if different chain lengths and types have differing biological activities.
Okay. If there are no more questions, Craig, thanks a lot for the talk and for answering all these questions. I don't know if, do you have some more time to stay in case anybody wants to have informal?
I have a meeting at noon, but or my noon time, but I'm willing to stay after. It's okay.
Okay. All right. So
I'll just stay on here. Thank you.
Yeah, thank you. We'll just stop the recording, and anybody who wants to stay for informal discussion is welcome to stay.